Good morning. Today I am going to present on the topic regulation of gene expression in eukaryotes. So in this we will see what is gene expression in eukaryotes, regulation of gene expression in eukaryotes. Moving on, next is steps involved in gene expression in eukaryotes and conclusion and references. So let's look on the question, what is gene expression? Gene expression is the process by which instructions in our DNA are converted into a functional product such as protein. When information stored in our DNA is converted into instructions for making proteins or other molecules, it is called gene expression. It is a tightly regulated process that allows a cell to respond to its changing environment. Moving on to the intro part in eukaryotes, like gene regulation in eukaryotes. It is more complex than compared to the prokaryotes. It involves more steps and interaction than in prokaryotes, and regulation can occur before transcription, after transcription, or after translation. Moving on, it is a combined gene expression can be defined as the combined process of the transcription of gene into mRNA, the procession of the mRNA, and translation into protein. So the main thing is that transcription happen, translation happen, and protein formation takes place. So here in this diagram you could see there's a gene in DNA which under transcriptional control we get our primary transcript, and then RNA processing control we get our mRNA. Then in nucleus and cytosol mRNA transformation happen, and where NRN transport control takes place, and then when it enters cytosol, translation occurs where translation control takes place and protein is formed. So it involves 6 steps. First step is transcription, second is RNA processing, third is mRNA transport, fourth is mRNA translation, fifth is mRNA degradation, and sixth is protein degradation. So which we will look further one by one. Here you in this diagram you could see DNA is after transcription and taken under by transcriptional control, which we saw before. We get our primary RNA transcript. After processing control takes place, the mature mRNA is seen. So mature mRNA is formed in this. This all happens in cytoplasm. After it comes out of cytoplasm, transport control takes place and mature mRNA is formed. Mature mRNA is divided into where translation control takes place by ribosome selection among mRNAs, and other half goes to mRNA degradation control. Translation, after translation we get proteins, and after degradation we get inactive mRNAs. The proteins formed after protein degradation control takes place, degraded proteins, we get degraded proteins, which we will discuss everything in further slides. So let us take a look on the steps involved one by one. First step, chromatin accessibility. The structure of chromatin can be regulated. More open or relaxed chromatin makes a gene more available for transcription. So when we saw that transcription is the first phase, but before that chromatin accessibility is required, which and this can be regulated. Moving on to the transcription. Transcription is a regulatory point for the many genes. Sets of transcription factor protein binds to specific DNA sequence in or near a gene and promote and repair its transcription into an RNA. Where it goes for RNA processing. RNA processing in RNA processing we have splicing, capping, and addition of poly A tail to an RNA molecule, which can be regulated and so can exit from nucleus. Different mRNAs may be made from the same pre-mRNA by alternative splicing. After RNA processing, we see to it RNA stability. The lifetime of mRNA molecule in the cystole affects how many proteins can be made from it. Small regulatory RNAs called microRNAs can bind, or miRNAs can bind to target mRNAs and cause them to be chopped. The next is translation. After RNA processing RNA stability, we will look on the translation. Translation of mRNA may be increased or inhibited by regulators. For instance, microRNAs sometimes block translation by their target mRNAs. And after translation, there is protein activity taking place. Proteins can undergo a variety of modifications, such as being chopped up or tagged with chemical group. This modification can be regulated and may affect the activity or behavior of protein. So the conclusion, we can say that gene expression is important because specific protein can be produced only when its gene is turned on. But it takes more than one step to get from gene to protein. And the process of building proteins is a key step in the gene expression pathway that can be altered in cancer. And these are the references. Thank you.